And basically, I mean, I'm just using this as a guide. I'll be putting some additional information up and doing some explanations that are there because we didn't really get very far in this. It was just mostly introductory things that I think are important. Just very quickly, the key issue, as you will see, just to make sure when we're going to get into the physiology of this, is the extraordinarily rapid rate of reproduction compared to us. Okay? We even on a cellular basis, we don't reproduce cells that rapidly, and we do. I mean, we replace, and you'll get to this in GI. We replace all of the cells in our stomach, small intestine, large intestine, billions of cells every day. But it's not extraordinary. It's not like an every 20-minute cycle. That's the thing with these microorganisms. They don't have a lot of other moving parts. They don't have to worry about the function that they're doing biologically. And all they are about is reproduction. So it's basically dedicated. That's what they do. They reproduce. Their waste products frequently are toxic to us, as we learn. They're chemicals. We call them toxins. Or when they attach or colonize to our cells, they cause damage. I mean, that's really what they do more than anything else. But the rapid reproduction is the big issue. So we'll get more into that, and you'll see it when we get into it. Large population, microscopes. Microscope guy is here today. He's working on microscopes. We typically get them done about once a year to try to make sure they're clean and that their surface would be the way to describe it. That's a really expensive piece of equipment, and you get that lecture in lab, I'm sure. I guess everybody's had lab by this. Yes, we're having it today, maybe. So who cares? Mushroom because you can see it. Again, what makes it microbes? We have to use a microscope to see them. Cannot by definition see them with the naked eye under. For instance, algae, as you can see up here. That's why I wrote it. They can be single or multicellular. A single cell you wouldn't see. The more multicellular it is, eventually you'll be able to see it. The worms that I don't do, the helminths, the single entity, and typically a lot of them, like a tapeworm, they keep growing and growing. The segments, a single segment you would not necessarily see. Eventually, as there are multiple segments, you likely would see that. So I mean, I don't want to give you. I want to give you some context when they do these kinds of questions. It's not a question I would use necessarily, anything along those lines. Okay? But I do want to emphasize these terms. Yeah, without, and I don't want to go crazy with the evolutionary biology and things like that. To me, because it's not an area of my expertise, I don't feel necessarily qualified. And I will always, if you haven't, many of you haven't before, I do not feel qualified to comment on the subject. I won't. Or as I will get to maybe a little bit today, when I have something that's opinion, I will frame it as opinion. Instead, if I'm teaching something that is factual, I will let you know. When I make a comment, as I might do today about COVID, that's a lot of that's my opinion. And inevitably what you'll glean from anybody who teaches you, you kind of get their spin on it. You'll know whether they are whatever class they're taking, whether they're this or that. And here you're going to hear my spin about vaccines, which I'm not necessarily all pro vaccines. Okay, that's my opinion from my soon-to-be 75 years living. Thank you. See, she inherently went. But the rest of you just sitting there going, see, in Yiddish, it's called being an Altakak, which means an old fart. I digress. So, they've been around, take it or leave it, for 3.8 billion. I didn't count. Okay. The big difference is eukarya versus prokarya. Because I phrase a lot of the questions that way. In eukarya this or in prokarya this. Eukarya, again, means, so that's all of these, eukarya or eukaryotic microbes. They're very much like ourselves. Okay, in the sense, and really the watchword for all of them is this. It's not just the nucleus. Nucleus just easily is identified. It's membrane-bound organelles. We're here. Some authors say, well, they don't have any organelles. That's incorrect. They just have no membrane-bound organelles. That's the difference. It's a layer of complexity. When you have organelles, membrane-bound organelles, you're compartmentalizing the work. It's a much more complex mechanism. It's, we talk about bacteria and viruses, I'm going to talk about their elegant simplicity. Not elegant because it's desirable, but from a structural standpoint, from an observer standpoint, why they are so dangerous or why they are so beneficial, is they're simple. And not as complicated to understand as maybe people have led you to believe that are there. So that's the big difference. Membrane-bound organelles, including a nucleus, that's where the carry-on, kernel of the core, comes first, versus pro or pre before the nucleus. Okay. The difference between bacteria and archaea, to simplify it, and we'll go into it much more when we, particularly bacteria, when we look in the next chapter, we really do bacteria anatomy. And it's extraordinarily important. When we're going to talk about the single most important two terms that you deal with when you deal with bacterial infections, and they're called gram-positive. It's a proper name, not the measurement. Capital GRAM and gram-negative. That's the ball game. That's what you're going to be doing in your lab. You're going to learn the staining sequence and how to identify them, the colors, etc., etc. So I'm preparing you for that as much as whoever you have. Most of them have Dr. Dress, who is much nicer than I am, and yes, younger. Yeah, just thank you. Bless you. He got a kid, I think, out of college and went in graduate school, went to college. Let's look at age table. So, bacteria found everywhere. The big deal in their cell walls, and all of these prokaryote by definition. Well, I made a mistake. This should be archaea. Thank goodness. Bigger. Bacteria and archaea. The big deal is they are single cell wall. Those are their major similarities. No membrane-bound organelles. But this is the big player. This is so important. This is unique. And again, using the term unique, I'm going to say so many times you're sick of it. That means it only occurs there. That's a distinguishing characteristic of bacteria. They contain peptidoglycan in their cell wall. Period. Is it the same amount, everyone? No. That's an important distinction. But it's always there. And they live everywhere. So those are the big things. What distinguishes archaea or archaea, however you want to, and this is the most you'll ever have to know about it. They don't have peptidoglycan. They have something similar. It's called pseudo-peptidoglycan. It's like it, but it's not. Not the same. And they only live in extreme environments. Those are the big features. So again, like weather today is not extreme compared to it. It may be extreme to us, but to archaea, you're talking about polar ice cap, bottom of the ocean. You get no place you want to go on your vacation unless you're a little crazy. Eukaryotic microbes, there's basically three classes. Algae. They can be single or multicellular. Again, a unique feature that no other eukaryotic microbes have, chlorophyll. They are, when you have chlorophyll, it means they are photosynthetic. And indeed, according to this text, and everybody's a little bit different, 70% of our oxygen planet-wide comes from algae. Remember, we're 85% covered in water. Algae are, by definition, single or multicellular. They have a cell wall. They're water-based, and they're photosynthetic, which means they have chlorophyll. And the chlorophyll, think of them as microscopic plants, almost. And it really, I mean, when you would look at a full cluster of algae, and there's other stuff that goes on, and they can be dangerous, like red tide. But we're 75% water covered, maybe more. The bulk of our planetary oxygen. Maybe not here when we're land-based, sitting here in, not near any major body of water. comparatively speaking. Our oxygen would come from there. But remember what photosynthesis is. It's fabulous. We take carbon dioxide and water, our two major waste products. We exhale carbon dioxide, we exhale water, we urinate out water, we defecate out things. Well, a fair amount of feces is about a third water. So we get water. Water's gotten rid of pretty easily. We have carbon dioxide. In the presence of sunlight, as the energy source, we're able to take that with the aid of chlorophyll and make effectively the food base for us, carbohydrates, basic carbohydrates, that organically we can tinker with, and environmentally and inside of it, so we tinker with to make all the stuff we need. And as a benefit, we get oxygen on top of it. We're a country. You should go out and thank a plant today. Please. I'm going to talk to you. Plants are living too. Moving on. And so I'm not worried about the other definitions that are here or anything like that. When we're going to get to it, the equivalent, not the equivalent to long term, what plays a pivotal role and what this PowerPoint goes on to talk about, because I kind of get a gist of what I want to tell you about from the first class and try to refine it a little bit. It's always been a little bit different. Basically the same content. We talk about effectively what bacteria plays. We talk about the roles. Microorganisms. They recycle stuff. They bring it back to our environment. They return elements like particularly nitrogen. And they're able to what they call fix. Fix isn't repair. It's short for affix. So we're gonna hear about fixing carbon and fixing nitrogen. Fixing carbon means taking, as plants do with chlorophyll, they take carbon dioxide, the carbon source, out of the air, and they staple it together by way of chlorophyll and sunlight. Create bonds to restore energy that we utilize for a variety of things. That's called fixing carbon, taking something inorganic, carbon dioxide, and turning it into something carbon. Bacteria, plants do that, algae do that. Bacteria also fix nitrogen. They take nitrogen gas out of the environment and nitrogen in the soil, inorganic nitrogen, and they affix it to organic compounds. And remember, amino acids, making proteins, nucleotides are organic compounds with nitrogen. Nitrogenated organic compounds. It's very significant. How do we recycle the material here? Effectively, what they are, they're called decomposers. The fancy name is saprobes. SAPROBES. They're typically based on land. So, and around here, we see it. How many times do you see it? Forever, particularly certain seasonal areas, we see roadkill. And you'll see it. And the next time you go past it, maybe you're seeing rigor mortis. And then all of a sudden, you're seeing what's left kind of turning kind of into mush as the proteins denature and it begins to undergo its decomposition. That is assisted by bacteria and fungi or yeast or mold. And what they're doing is they secrete enzymes. The enzymes gobble it up and they utilize that as their nutritional source. In the process, their waste products are returning things that we need to build stuff back to the soil, back to the environment. So, I mean, this is just some of the many, many roles that microorganisms have. So these have a cell wall, but they don't have chlorophyll, which is odd. So they're sort of that in-between guy. They're not a plant or plant-like because they don't have the chlorophyll, but they have a cell wall. So they're very interesting. They do cause us a lot of problems. Fungi cause us a lot. Many you're going to run into them. If you haven't had them, you're going to see fungal infections. They're very more common than you think. And I mean, for instance, I just saw an article about something that because when they're bad and they get into your lungs, they're particularly dangerous. They're having an outbreak of histoplasmosis, which we'll get to when we do respiratory disease. That's the last third of the course. Histo is called spelunker's disease. People go caving and things like that. So it's particularly true with moist environments, it's moist caves that we have around here, but we see it more in southern states with swamps and things like that, and bats frequently are associated in their habitat with that. And there's been an outbreak of this called histoplasmosis. it isn't a bad idea, as you whatever you get your news from or look at what. Every time I see a food recall, look at it. Every time I mean, if you want to get fancy, you can go on the CDC's website. They have a weekly report. They will tell you about not just food recalls, but outbreaks. Whether it's Listeria monocytogenes, which is nasty, generally comes from animal fecal contamination in our food supply. That is in water, and the way the food is processed, and things like that. Those are kind of interesting. And so when I see something interesting, I try to bring it to you. But it's, I mean, if you're going to be in the healthcare tree, most of you are planning to be, you need to be wary of these things. You want to know if there's an outbreak of something going on around here? I mean, I think everybody, if it didn't hit you in your family or within your orbit, this flu sucked. My wife was really sick for 2 days. I mean, ordinarily she's a pretty healthy person. I'm the one who has to get taken care of. She's younger and better looking than me. Much younger and much better looking. Nevertheless, so I don't want to worry too much about this chart, such as it is. I think I covered not much there. Okay, single cell eukarya. We're eukarya. So we're different because we're macroscopic. Animals and plants are not in there. Are we part of it? For sure. The classification of kingdoms was there. The big difference is something I touched on a little bit. I remember making a big deal of it when we were here on Wednesday, was the ribosome. Remember, the way that the domain system was, so this is a domain, this is a domain, and eukarya is a domain. And these are just subdivisions of it. The domain, what's interesting about this, they found in the ribosomal RNA, and we're going to go over it again, so you don't have to worry about it. They're called 70S. The S is a proper name for density called Svedberg unit. You're going to see, and again, that is in the ribosomes. It's part of ribosomal RNA. And you might remember me going through that whole thing with antibiotics and allergic to penicillin and stuff like that. That's what a lot of antibiotics target: protein synthesis. They target the ribosome. Why it's useful but not perfect, and this is what the domains with Weiss, who was the author who came up, the research came up with the domain system back in 1978. I didn't even hear about it. I graduated my doctorate in 1977. We still had kingdoms. It's a lot easier. I figured he came away. Some people like to publish a paper to make life more complicated. But in eukarya, it's 80S. It's a denser ribosome. The interesting thing, and we're going to learn about this, it's really important, is something, it's as though our more advanced cells, called endosymbiosis, came from, it's almost like two simpler cells one swallowed the other, and rather than eat it, kind of incorporated it, found it useful. That was really the start of membrane-bound organelles, and we'll go over the science. So this is what's coming in the future. And the interesting thing is that in eukarya, mitochondria have 70S ribosomes. That's why there's side effects with a lot of these antibiotics. Big problem with antibiotics that target protein synthesis side effects. Big problem with penicillin targeting peptidoglycan allergies, comes from mold. I mean, does it preclude side effects or preclude allergies on any way? No, it does not. Allergies are very interesting. I don't know how much you're going to get about it in immunology, but I mean, you can take a medicine or you can take, ingest a substance for 50 years, and all of a sudden the next time you take it, you're allergic. I love strawberries. I can have it, and all of a sudden, bang, you're allergic to strawberries. Your immune system follows its own mind. I had that with a lady. First question I asked, diabetic lady, I can tell you her name. I treated her. She's an old nurse. She's no longer with us. Titusville, PA. And she was like that, the nurse everybody knew in the hospital because she was there for 1,000 years. And I saw her, she had an infection in her foot. And my first question, are you allergic to penicillin? It was already on our chart. She goes, no problem. I gave her a prescription for amoxicillin. And in those days, the local paper up in Venango County would tell you who's in the hospital. I was practicing all this. I saw her name. I'm like, oh boy, am I going to get sued? So she called me in a huff. She goes, that penicillin you gave me put me in the hospital. I talked to her physician. I said, but you told me you were allergic. Well, I never was, but it must have been the one you gave me. And she did fine afterwards. And I had a very good relationship with her medical doctor, who was taking care of her diabetes. But that just goes to show you, she was in her 70s. I mean, I treated her for at least 20 years, because I treated her when she was in a nursing home, probably past 90 years old. So always expect the unexpected. The fact that you take a history and it says allergic to penicillin or not allergic, you always have to be aware of it. If they take a medicine, any medicine can cause any side effect. First thing when somebody has a problem and they're on a new medicine, check the medicine first. Take that away and see what happens. Assuming that you have that option. All sorts of little things, because I've been on the business end of some things like that. You got to be very. Moving on. So, and like us, highly complex eukaryotic cells are bigger. Compared in the microbial world, bacteria massively predominate. I mean, in your large intestine, you have 10 times as many bacteria lining your colon than you do have cells. You have billions and billions of cells. You have tens or hundreds of billions of bacteria. I mean, it's just filled. And they're your friends. Where E. coli comes from, E. coli is not bad guy. E. coli is something that helps you break down a lot of things in your gut, and basically that's why we take probiotics. Probiotics are good bacteria that either have to replace bacteria that's gone. So maybe you had I got food poisoning and I couldn't stop going to the bathroom. And what are you supposed to do? You have to replenish that. So whether you're eating yogurt, taking probiotics, whether you're anything like that, buttermilk, if you're inclined to have that or use that, there's lots of things. And you'll find that we talk about antibiotics, but certain antibiotics are going to cause digestive, and the most common side effects are gastrointestinal. I would have patients start now. You need to take probiotics. You need to take yogurt. Take this with yogurt. So you hear more about that as I come to talk to you. That's my area of expertise. I'm not nearly as bright as some of the people who are going to be teaching you. Academically, they're sharper than I am, but I have a lot of clinical acumen. You all know, a very good memory. I was impressed that somebody knew who the Four Seasons were. Very impressed. Yeah, the only one I don't like is winter. It's a joke. They didn't get it. Just texting my sister, who her husband retired down in Charlotte, North Carolina. I said, how much snow are you? They're expecting an ice storm. They figure they're going to be without power. They're not well equipped. You're supposed to get like a half an inch of ice. I have no idea. She's older than I am. 5 years, 7 months, 1 day, and 1 hour older than I am. Offhand. Okay, so ubiquitous. When I teach the lab, and I've taught this lab, I can't even tell you how many times in different schools. Ubiquitous means a really great word. It means they're everywhere. And microorganisms are everywhere. They're all over you. They're all over your clothes. They're all over the carpet. They're all over your shoes. Microbes that can kill you. There. We'll go into a reason, when we go into the historical figures, we'll go into a reason why you always have to be concerned about tetanus. Because it's everywhere. It's on the bottom of your shoes. It's in this carpet right now. And bad news, the way you find it in there, it lives effectively forever. It lays dormant forever. It's called an endosperm. Indeed, when we sterilize something, the definition of sterilization is the destruction of all microbial life, and we get to this, we have a great unit on that, including bacterial endospores, of which 10 is. We're just starting. So they're everywhere, ice caps, extreme environments, inside plants and animals, on top of us, everywhere. There are areas that are we call sterile inside the body. They're not allowed to have those. Okay? So when you're talking about when an appendix bursts, that appendix pours out all those aforementioned bacterial organisms that normally would seed or be in the colon, and they pour them out into the abdominal peritoneal cavity. And that's the thing called peritonitis. That's an immediate life-threatening problem. That's a trip to ICU. That is surgery. Right now, emergency. So someone gets like a bullet wound or a knife, a sharp trauma, any kind of trauma that does that, or the burst appendix. And then you have the bacteria where they're not supposed to be. That's a otherwise sterile environment. All of these things are terribly relevant, and you will begin to understand, and I hope to kind of put the package together clinically. I'm actually doing a better lecture with you guys than I did with you. It's like, don't tell Victoria. Theory, all they're telling you, when you see the term theory in a science class, it's not like, I have a theory. No. This has been around for a long time. People have tried to disprove it. It's just this side of a law. So you can get Newton's laws, and energy laws and stuff like that. This is close. So evolution's, I'll buy it. Again, I don't have enough expertise to teach it, but that they're just telling you that it's not a random guess. Who cares? This is the whole idea that I was talking about. Energy flow, nutrient flow. So we have to bring back other resources. Photosynthetic organisms are providing. Eventually we're going to go. A lot of our food source is going to come from these areas. There's a ton there that's going on. Okay, and so long before plants, we had photosynthetic bacteria, photosynthetic microbes, and there's some exotic bacteria that we don't really deal with that you would probably find closer to archaea that are photosynthetic bacteria, which is so that we call it pigment. Very, very interesting. And again, and we start off without them, as you will see, because they have this here. Here's the photosynthesis, light fuel, that's the energy source, and we are able to harvest it. Carbon dioxide and water, they should have incorporated that. Again, it's PowerPoint, no editorial review, they make mistakes. And oxygen is a side benefit from that. When the planet was first born, for lack of a better term, it was an oxygen-free atmosphere. Only until photosynthesis evolved, they had different kinds. Anoxygenic was before plants, and really, so originally the atmosphere was not oxygen, and gradually over this, whoever author you read, how many billions of years it transformed. And that's the best thing here is the 70%. Main force that drive the structure content of soil, water, and atmosphere. I'm not sure about this. And again, PowerPoints sometimes are erroneous. I can buy the carbon dioxide. I can buy the methane. Again, when you're decomposing things, that's what comes of it. Those things. Nitric oxide, I'm not so sure of. They may just be talking about nitrogen gas or nitrates or nitrogen particles. I'm going to have to take a look at that and see. Again, they're talking about what I call fixing nitrogen, taking nitrogen gas and stapling it, so to speak, to organic compounds. Yeah, bacteria. Okay, most abundant cellular organisms in the ocean, and again, viruses are the most abundant inhabitants. Again, viruses are not organism. This is where terminology, and this is where you can, everybody knows this. On a test, you can come up and ask me. Okay, I may not be able to clarify the answer. I may say I can't give you more information, but at least you have the option. Licensure exam for the word organism, that means a living thing. If they use the term entity, whatever they used here, inhabitant, that is the implication that it includes both living and non-living. Again, viruses ain't alive. to say it mildly. Go back into New Jersey and New York East if you like. Get it. So bacteria and fungi live in close association with plants and the system. We're all working together. Yet they're sitting all over you. That bad? No. They're covering up receptors that bad microorganisms can attach to. They're gobbling up nutrients. They're breaking down. When they break down, they produce they break down some of the cell membranes and cell walls and produce fatty acids. We have what's called an acid mantle. Most microorganisms don't like that. Our perspiration is salty. That's why we're covered in staphylococcus. It's called Staphylococcus epidermidis. Staphylococcus you find on the skin. Staph aureus, which is a pathogen, is on there too because we grow it in our nose. That's what normally it grows. So it's every time you cough or sneeze, there's some on there. Why does it survive? It loves salt. Most bacteria don't. That's why salt forever is used as a preservative. It's the oldest preservative that we knew. Maybe honey and vinegar to a certain extent probably were at the early onset, but salt was. You heard this before. They paid the Roman soldiers in salt. That's where the word salary comes from, to do with salt, because they could preserve fruits, vegetables, meats with salt. And that was the expression, they weren't worth. He or she was not worth their salt. Love this. If nothing else, you'll have enough obscure facts to compete in like a trivia contest. At one time I was undefeated in those, but not anymore. Now, anything past 1980, I have no idea. Pop culture references of the 90s. So, yeah, freaking everywhere. This is the interesting part. This is where we start to look at fungi and yeast, bread, alcohol, cheese. These are fermentation products. The most common of these, and we will look at it, are generally in the considered yeasts. And so bakers and brewers yeast are the same thing. We'll have it in print down the road, and I'll write it down here if you want to. Saccharomyces. The saccharose means it uses carbohydrates. Myces implies it's yeast or fungal, and the last name is Cerevisia. Many of you would know that the Spanish word for beer is cerveza. That's where and we use this whether you're making sourdough bread, whether you're making pizza dough. We go get those little packets of yeast. Boom, you're going to use them in the lab, and voila, put them in water. Those little granules have thousands upon thousands of microscopic yeasts in there, and the water releases them. Give them a little sugar to feed them. They do what's called blooming. If you're like me, I love to cook. And the next thing you have is you can make bread that way, pizza dough that way, however you want to make it. Okay, and all those cheese the same way. Different cheeses use different, typically bacteria. Whether you're using like making a blue vein cheese, Swiss cheese is a particular bacteria, and imparts different kinds of flavors depending on how they make it. So, and it depends on. And so there's all different areas. That's why different cheeses taste different if they come from different animals in different regions. That's what, Pecorino Romano doesn't taste like Parmesan Reggiano. Parmesan is a cow milk cheese. Pecorino is a sheep milk cheese. Little cooking stuff never hurt anybody. If you want a great recipe for like a fancy dinner, I have it's like a 3 cheese wrapped in pastry. Oh my God, was that the hit of my daughter's inevitable white elephant Christmas Eve party for all of her in-laws. I just come to cook. So wounds, and I looked this up. I mean, I knew that they, when they wanted to gobble up dead tissue, they would put maggots. The maggots would, in ancient times, they gobble up this leeches to pull. Leeches would suck material out of it. It would be like drawing something. Making in the old days, we used a thing called drawing sap. They don't use so much, but leeches you would put on there, and there were bacteria on the leeches, and they would compete against the bad bacteria. They would cover things in honey and vinegar. Vinegar is very acidic. Honey, the sugar, even though you think it might be counterproductive, I mean, it alters the osmotic area. It alters the amount of pulls moisture out. Well, there's all sorts of different things. So microbes have been used directly and indirectly with this kind of stuff. Modern wise, well, we basically don't use that so much. I don't know enough about the precious metals, and we use modified microbes all the time for oil spills, industrial waste, water treatment facilities. I mean, it's a really good field trip. I don't know if they still do it in our environmental science classes. They used to have a field trip to go down to the water treatment facilities. And you see all ultraviolet light and filtration and different things they run them through. Very fascinating. We have the luxury here, as we do throughout the country, more or less, barring unforeseen circumstances, of having a pristine water supply. That's why we don't have to worry so much about protozoans. Protozoans. This is when somebody, if you have know somebody in a lot of, we have mostly city water, but we're not too far away from people who have well water and septic tanks. You worry about amoeba, amoebic dysentery. You worry about giardiasis. Those are protozoans that can be potentially deadly. We'll talk a lot about protozoans. You might remember me saying the single most deadly organism, by numerically speaking, in sub-Saharan Africa is a protozoan. It causes malaria. People always think it's AIDS and stuff. We'll get into all those types of things. I hope I have time to do this last part. Yeah, here it is. It's hard to me. Let me explain it to you. Rather than, because I don't have a lot, they just have a board there. I am not taking down my periodic table of elements. You know my affection for that. It makes this a science classroom. Feel better. Science. Okay. I will. The next time we're physically on site, I'll draw it. All of this genetic engineering and recombinant DNA, not so much even the bioremediation, because we genetically engineer the bacteria to gobble up oil spills. All that stuff comes from microorganisms. And we'll go into, we look at bacterial genetics. Bacteria make enzymes that we don't. Well, fancy restriction. What they do, their job is chop up DNA. So we learned about this. We took certain bacteria and we harvested restrictions. Remember, DNA, as big and giant it is, it's relatively simple because it's a simple language. A's, T's, G's, and C's, right? It reliably, consistently chops the DNA. So how do we begin to be able to do crime scene testing, for instance? That whole Ancestry.com, 23 it all came from bacteria because restriction enzymes put it with our DNA and chopped it up into manageable fragments. We could do testing with it, PCR testing, things called RFLP testing. And what it would do, we could get a genetic picture. Years ago, we couldn't. The best we could do was analyze the chromosomes to look if somebody had Down syndrome, trisomy, karyotype. So we took this further. Most bacteria have different kind of DNA. They don't have pairs, they have big circles. A lot of bacteria have these tiny circles. Fancy name we're going to learn about called plasmids. And the plasmid up here is a little circle of what's called non-essential but useful DNA. It typically carries resistance. Okay, what's up? So if you hear about MRSA, most of the time MRSA is methicillin-resistant Staphylococcus aureus. That plasmid can move from bacteria to bacteria. The magic of bacteria, it can change something we want to do. Magically, I throw away my glasses. Magically, could I all of a sudden be a good athlete? I have a vertical that I told you about before. That's it. Done. Your particular. It's imagine if, let's say, she had perfect health and perfect vision. She could touch you and you could touch her, and magically all of us would have that as soon as we touch the next person. That's what bacteria can do. One MRSA, by way of this thing called a plasmid, because there's a mechanism to shift plasmids, can change a population to all MRSA overnight. Overnight. No wonder antibiotics didn't work. It's mutating. It's a mutation. It's genetically unstable. This is what genetic engineering, we took this, and we were able to take those enzymes, chop the plasmid, make a piece of DNA, and magically slave the bacteria that was receptive to this, the so-called recombinant DNA technology. We could take this plasmid, take it to a bacteria that didn't mind accepting new material, and resequence it, rechange it. Magic. And now all of a sudden that bacteria would make stuff that's useful. When someone's going to have a baby, they're normally producing, which you're going to learn about in endocrine, called oxytocin. It's a hormone made by the posterior pituitary. It's a very short chain amino acid. So in the hospital, we make it genetically. It's called pitocin. To induce labor. Oh, I'm going to give a shot. We're going to give a shot of pitocin. We want to get the process started. Okay. The first genetically engineered organism and the agent with a chemical that it produced insulin, human insulin. Insulin originally came from pigs because they were abundant and inexpensive, but there were a lot of people who had allergies and a lot of people who had religious reasons to using that. When I started in medicine, remember a long time ago, we were really just utilizing bovine, various kinds of bovine insulin from cows. The same problem, not that there weren't nearly as many religious problems, but there were still allergies. And in 1977, the first genetically modified made with insulin. We were able to identify chopping up the DNA sequencing, create it in the laboratory, enslaved bacteria, probably made by Lilly out in Indianapolis, to make human insulin. Now, basically anybody on insulin is human insulin, less likely to be. Hepatitis B immunizations, insulin. Hepatitis B immunization is genetic engineering. A lot of vaccines, genetic engineering. All of those things. I'm trying to acquaint you in this long-winded manner of all the good stuff it does. Most of them are good guys. There's only about 2,000 that are bad guys, but there's a ton of infections. I don't want to start this next slide. I have some issues with this, particularly the COVID thing, and we'll talk about it next time. I don't want to. We'll cover COVID. I have a pretty good understanding of it. You're going to get a lot of my opinion about it. I'll tell you the same thing I told the other class. I'm more likely one of your older professors, despite my youth. And only about 3% of us did not get the COVID injection. I won. No injection, no boosters, no. That's it. I mean, so I'm framing it that way so you understand my spin. I'm going to be as honest with you what I believe to be true, and I'm not dismissing the people, how many people are killed. I'm dismissing the number. I'm not dismissing the severity. We'll talk about that in the weeks to come. Thank you very much. Have a great. Be safe over the weekend. Hey, look, if you're not comfortable coming to class and they decide to have class, which I doubt, don't. I don't want to see anybody get hurt. I'm not that important.